Good morning. It's March 10th, and this is your daily brief in science. Here's everything you need to know. Roche disclosed that its Persevera Phase 3 trial of giredestrant with palbociclib for frontline estrogen receptor-positive human epidermal growth factor, receptor 2, negative metastatic breast cancer did not meet its primary endpoint of progression-free survival versus letrozole plus palbociclib, though a numerical improvement was observed. The company says it will continue developing giredestrant, including exploring combinations with CDK4/6 inhibitors in the adjuvant setting and pursuing other trials such as EVERA, lidERA, PIEZO1, and HEREDERA. Analysts pointed to the trial design and its comparison to rival SERD camizestrant as a factor in sentiment, with camizestrant viewed as structurally advantaged in some analyses. Broader market sentiment shifted toward caution in the oral SERD space following Roche's results. Investors are watching Roche's patent horizon, with some estimates suggesting peak annual sales around CHF 1.2 billion if the program succeeds, though earlier forecasts had giredestrant potential at up to CHF 5 billion in sales by year 2030 in a favorable scenario. Some analysts see a buying opportunity for palazestrant, arguing its stronger antagonism and pharmacokinetics could enable it to outperform giredestrant in similar settings. And Stifel notes palazestrant's advantages might drive best-in-class status if differentiation is achieved. Jefferies raised Roche's price target and upgraded to buy, citing possible advantages in camizestrant's design that could affect Roche's standing in the SERD space. The setback raises questions about frontline efficacy and commercial viability of oral SERDs, especially as competitors like AstraZeneca's camizestrant advance with their own data and strategies. Camizestrant's potential clinical impact, particularly in ESR1 mutation patients, keeps it in focus with an FDA advisory meeting planned and adjuvant data anticipated next year. Competition remains intense in the oral SERD field, with AstraZeneca's camizestrant and Olema's palazestrant among rivals pursuing first-line and later-line opportunities. A whole cell model now lets us predict nucleotide metabolism, DNA replication, and ribosome biogenesis all at once, offering integrated views of core cellular processes. Instead of simulating every atom, the model averages molecular dynamics to time the cell cycle, acknowledging a limitation but delivering strong predictive power. This work comes from the National Science Foundation's Science and Technology Center for Quantitative Cell Biology at the University of Illinois, which aims to advance computer vision approaches to fundamental life processes. Researchers note the difficulty of simulating moving three-dimensional cellular components and sometimes render interior elements invisible to reveal events like chromosome threading. Co-authors include Angad Mehta and Taekjip Ha, with contributions from Zane Thornburg and Andrew Mayton to refine simulations and address real-time cellular events. The study was published in Cell, with project details available through qcb.illinois.edu and JCVI resources. Collaborations with Harvard Medical School and the J. Craig Venter Institute, and the use of NSF-supported Delta Computing Resources at Illinois, supported the project. The team integrated experimental data from multiple institutions with extensive computational modeling to map thousands of molecular interactions and reactions. Interdisciplinary collaboration spanned computational biology, chemistry, physics, and experimental biology, leveraging data from Harvard, Illinois, and NSF facilities. Advances in multiscale modeling reconcile mechanical and biochemical interactions, enabling exploration of cellular responses to stress or mutation, and informing synthetic biology and minimal life research. A dual GPU strategy accelerated simulation. One GPU handles chromosome replication, the other handles metabolism and ribosome assembly, completing a full 105-minute cycle in 6 days of computer time. The modeling relies on high-performance computing and optimized simulations to manage thousands of molecular interactions, with replication requiring dedicated computational resources. A new study in Science Advances spotlights a mechanical route that could shape early breast cancer intervention. Researchers identify the PIEZO1 Rho-ROCK axis as a promising target to limit growth and invasiveness through a concept called mechanotherapy, therapies aimed at disrupting the tumor's mechanical signals. The work suggests that brief mechanical pressure can reprogram cancer cells via epigenetic histone changes, creating a lasting memory that sustains aggressive behavior even after pressure is removed. PIEZO1, which is more abundant in breast cancers than in normal tissue, activates a calcium influx that triggers the Roche pathway, boosting cell movement and tumor growth. The study shows that brief compression of cancer tissue markedly increases growth and promotes epithelial mesenchymal transition, a process linked to higher invasiveness. Blocking PIEZO1 or the Roche pathway with drugs prevents this compression-induced aggression, identifying these components as key mediators of the mechanical response. The research also demonstrates a durable epigenetic memory, where histone modifications propagate the aggressive phenotype over time, beyond the initial mechanical stimulus. This memory highlights how mechanical signals can have prolonged effects on tumor behavior and progression. The team points to mechanotherapy as a way to identify patients at risk of aggressive cancers and to slow tumor progression early in the disease. The study, published with DOI 10.1126/SciADV/AEB1271, received funding from the Hospital Research Foundation Group, Australian Breast Cancer Research, Worldwide Cancer Research, and the Australian federal government. Overall, the findings establish a mechanistic link between tissue pressure, PIEZO1 activation, and downstream Roche signaling as a driver of breast cancer growth and invasiveness. A potential game-changer in autoimmune therapy moves forward as China grants IND clearance for SKB575 with HBM 7575, a long-acting bispecific designed to tackle upstream type 2 inflammation in atopic dermatitis. This milestone clears the way for clinical development under the Kelun Biotech and Harbor Biomed collaboration, leveraging Kelun's ADC and DC platforms with Harbor's Harbor Mice and HBIC-EHBIC platforms. Harbor Biomed's Dr. Jingsong Wang says the pair aims for best-in-class status by targeting TSLP alongside an undisclosed second target, delivering a dual mechanism with extended half-life for dosing every 3 months or more. Kelun Biotech operates as a holding subsidiary focused on innovative biologics and small molecules, with multiple OptiDCAD platforms in development that support the collaboration's drug development pathway. The IND aligns with Kelun Biotech's expansion into autoimmune diseases and fits Harbor Biomed's strategy to push next-generation antibody therapies for inflammatory conditions, including atopic dermatitis. Leaders from both companies emphasized patient convenience and durable disease control as goals, with subcutaneous administration and a prolonged dosing profile in sight. The collaboration's shared approach combines Kelun Biotech's broad development program with Harbor Biomed's discovery and development support, aiming to disrupt upstream inflammation rather than just treat symptoms. Atopic dermatitis, a chronic inflammatory skin condition, affects a large patient population and often lacks durable control with current therapies. The joint effort positions SKB575/HBM 7575 as a potential safer, longer-lasting option if clinical results meet expectations, though details on the second target remain undisclosed. NMPA's IND clearance confirms a pathway for a long-acting bispecific antibody trial in atopic dermatitis that targets TSLP and an undisclosed antigen, marking a notable step in this collaboration. The gut microbiota plays a significant role in cancer immunotherapy outcomes, especially with PD-1 and PD-L1 inhibitors, by shaping anti-tumor immunity through microbial metabolites and interactions with immune cells. Clinical data show fecal microbiota transplantation can restore treatment responses in some advanced cancer patients and may reduce immune-related adverse events. Beneficial microbes like AKKERMANSIA mucinifila, BIFIDOBACTERIUM, and LACTOBACILLUS promote anti-tumor immunity by producing metabolites such as short-chain fatty acids and tryptophan derivatives that affect T-cell differentiation, antigen presentation, and T-cell exhaustion. Integrated evidence from preclinical studies, clinical trials, and multi-omics analyses indicates commensal bacteria enhance immune activation, treatment sensitivity, and can modulate toxicity. A comprehensive review combines diverse data to demonstrate how commensal bacteria shape immune activation, treatment response, and adverse events. Emerging microbial biomarkers, alongside metabolomics and machine learning, show promise in predicting immunotherapy outcomes. Looking ahead, synthetic biology approaches, including engineered bacteria with safety switches or patient-derived strains, are proposed to deliver targeted immune modulation within tumors. Beyond cancer, microbiome immune insights may influence therapies for autoimmune and inflammatory diseases, positioning the gut microbiome as a controllable therapeutic platform for precision medicine. Microbiota-guided immunotherapy could reshape oncology by enabling dietary modulation, probiotics, microbiota transplantation, engineered live therapeutics, and AI-assisted microbiome modeling to tailor treatments and reduce toxicity. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.